Thank you everyone for coming today. We have Professor El-Kebir to talk to us about computational biology, which is what this presentation is going to be about. So take it away.
Thank you. Thank you for the introduction, and also thank you for the invitation for having me talk here. So today I'm going to be talking about computational biology, and I also will touch, after introducing basically what this field is about and how it's related to computer science, I will also start talking about my own research a little bit, just to give you a flavor of what you can do as a computer scientist in this field. So let's get started. So computational biology, or oftentimes you'll also see you'll see it referred to as bioinformatics. It's an interdisciplinary field that develops and applies computational methods, and their goal is to analyze large collections of biological data, and the ultimate goal is to make new predictions or discover new biology. So this definition comes from Nature. So Nature is one of the top journals, in the sciences, and they have like this really cool, resource where you can look up definitions of fields and common concepts. So they are basically defining CompBio and bioinformatics. There's a couple of things that are important here. So it's about computational methods, so developing new computational methods, and the goal is to discover new biology, and you do that through analyzing biological data. The key connecting pieces is biological data, and that's actually also related to kind of like what happened in biology. So until late 20th century, biology was very bottom up. So what that means is you would look at one protein, for instance, or one component of a big complex system, and you would try to understand this from this small component, and then you would work your way up. And the classical way you would do this is you would formulate a hypothesis and then try if you can actually validate it or refute it. So it was mainly hypothesis driven, and it was really bottom up. So you try to understand this big complex system by understanding individual pieces of the system. However, now with all this sequencing technology that we have, high throughput technologies, it completely changed the paradigm. So what happened is it's top down. So what you're doing is you're looking at a snapshot of this complex system in two different states, healthy, diseased, and then you're going to try to identify what are the differences. And from knowing those differences, you can actually start formulating and generating hypotheses and validate it. So it's really completely turned around, upside down. So now it's really top down. And in order to do this, you need computer science, because there's lots of data that you have to interpret and analyze. So let me just bring this point home. So what I'm showing you here is the plot that every person in computational biology is very familiar with, and it shows you the cost of sequencing one human genome. And as you might know, the human genome was completed in 2001, and the cost actually it cost about $3 billion. In terms of like total research expenditures to actually get this first draft. And to redo this first draft with all the knowledge gained at that time, it would have cost $100 million. And what you can see over here is how this cost dropped exponentially. So in white, I'm showing you Moore's law. And you can kind of like see, nowadays we're down to, $1,000. And it's actually about $500 these days. And something interesting happened here. In 2007, there's like this super exponential drop. And that's actually because of a new sequencing technology. So let me just tell you a little bit more about that. So when I say that we're able to sequence a genome, it's actually not what you think it is. So when we're talking about a genome, it's a very long sequence. So we're talking about billions of letters, billions of nucleotides, A, T, C, G. However, when you do next generation sequencing, you don't read out this genome from start to end. Instead, what you get are short pieces of the genome, fragments, or actually they're called reads. And reads are orders of magnitude smaller than the length of the genome. So typically with next generation sequencing, so the sequencing technology that caused this really steep drop in cost, the reads have a length of 100 nucleotides. And compare this to the reference to the human genome, which is 3 billion letters long. So this is orders of magnitude smaller, and you basically get these short pieces that you need to put together into a bigger genome. So the key point here is, no technology exists that can sequence a complete genome from end to end. So we always get these short pieces that we need to assemble. So in order to make sense out of this data, you need to use computer science and algorithms. It's absolutely necessary. There's no way around it. And that's actually where, why this is such an exciting field. So for me personally, what I like a lot is it's very interdisciplinary. So as a computational biologist, if you study in this field or work in this field, you're exposed to different disciplines, including biology, computer science, mathematics, statistics. So you need statistics to make sense out of different error models that you have because of the different technologies. And this is really fun. So you basically get to work in different fields simultaneously. And yeah, I always show this in class. So this is a bit of a dated ranking. So it actually shows you a ranking of jobs in terms of how much employees appreciate their job. So best jobs to worst jobs. And when you look at the top 10, about 6 jobs in here that are all related to computational biology. So it's pretty cool. It's interdisciplinary. You get to do many different things. And the other thing that I find extremely exciting about this field is, I just gave you a little taste of a problem, a computational problem that you have to solve. You have to make sense out of these short reads and assemble them into a longer string, a longer sequence. So that's just one type of problem, but there's many more in biology. And actually, there's this really cool quote from Professor Don Knuth, who's like a really famous computer scientist. And what he says is the following. So he can't be as confident about computer science as he can be about biology. And here's the key quote: biology easily has 500 years of exciting problems to work on. So it's extremely cool. So essentially, what this really means is you have classical computer science problems like traveling salesman, like shortest path, and there's all kinds of graph problems. But the cool thing is when you start looking at computational biology, you typically get a little twist on a classical computer science problem. So there's going to be one different constraint that you need to look at, or maybe the weights are completely different on the edges of the graph, or there's something else that makes it a little bit different from problems that you would learn in an algorithms class. And that's really cool. So essentially, you get to do new computer science because of biology. So that's basically what is what Professor Don Knuth means here. So there's a lot of computer science problems that are inspired by biology. So biology leads to new computer science as well. So that's really what I like, and that's also what we do in the group. So we really try to identify new computer science problems for to actually understand biology better. But the cool thing is this translation from, what is the biological question can be formulated as a new computer science problem, and then actually understanding this computer science problem. So you start thinking about is it hard? Is it easy? Can I solve this efficiently? Or do I need to use heuristics? So there's all kinds of algorithm engineering that you have to do, but also understanding the problem that you just posed. You have to analyze its complexity, and that's just straight up computer science. Good. But the other important point that I want to make is actually getting this problem statement. You need to have a solid, grounding in biology. You need to be able to understand the biology well enough in order to identify the key pieces that are relevant. So you need to be able to arrive at a computational problem by understanding the biology well enough to understand, which details you can leave out and what are the important pieces that you need to include. So and that's something that a standard computer scientist won't be able to do, but a computational biologist would be able to do that. So this formulation of a new computer science problem, and then once you have the computer science problem, you can give it to any professor here in the department, and they will be thinking about it and come up with a solution. But this initial translation of biological question to computer science problem, that's also where a lot of innovation is necessary, and a lot of thinking is necessary. Good. So let me tell you a little bit about yeah, a course that I'm actually teaching this semester. So this will give you a sense of, so I just want to go over the topics that we're covering in this course, and that will also give you a brief taste of what this field is about. What are some of the fundamental problems that we're looking at? So the course that I teach, it's CS 466, Introduction to Bioinformatics, and you absolutely, so there's no prerequisite on biology. You don't need to know any biology at all. So we're just going to learn it on, as we go through the lectures. I will introduce the relevant biological concepts, but I don't expect any knowledge of biology. And the prerequisites for this course, so this is actually an upper division course. So it basically, you need to be familiar with, discrete math, asymptotic running time, some typical data structures and algorithms. So essentially, the prerequisites are CS25 and all its prerequisites. Because there's a lot of computer science involved. So we're going to be looking at algorithms. We're going to develop, study current algorithms and analyze them. So you need to be able to, to understand mathematical proofs and also be able to formulate, proof, theorems and lemmas. So that's part of it. Good. So let me just go over the course topics, because that will also give you an introduction to the field. So the first key topic that we cover is sequence alignment, and the underlying biological question is about how do you compare two genes or genomes. So for instance, when we look at a human genome versus a mouse genome, they have different lengths. So the human genome is 3.2 billion letters long, and the mouse genome is 2.8 billion. So you want to be able to somehow compare these two different sequences. And when I say compare, you want to identify what are pieces that are similar and what are pieces that are different. And this is actually a string comparison that you want to do. And you can do this in different ways. There's different flavors of problems you could look at. So that's it's one of the fundamental things that you need to do. Sequence alignment. So that's the first topic. The second topic I already alluded to. So when you do sequencing of a genome, you get short reads. And you want to assemble these short reads into a larger genome, into the complete genome. So how do you put all these short pieces back together? And that's called genome assembly. How do you assemble genomes? And that's also a very interesting topic. And the key notion here is that you essentially have, so instead of looking at a single genome, you look at many genomes simultaneously. So you have many copies of the same genome. So you look at multiple cells and they have all this, they have similar genomes because they come from the same organism. And you basically get reads from roughly the same regions, and they will be overlapping. And so essentially it's like a puzzle. So you want to identify how to piece together reads that overlap, where the suffix of one read overlaps with the prefix of another read, and you want to put them together. And this actually becomes a very cool graph problem. So that's the second course topic. And then we also spend a lot of time thinking about evolution. So what do I mean by that? So, essentially, pretty much everything that we are is encoded in our genome. And you could start thinking about if you're able to compare different genomes from different species, you're going to see similarities. And the more similar two genomes are, that tells you something about divergence, like when did the two different species diverge. So the more similar they are, the more recent this common ancestor actually is. And you can actually represent this whole evolutionary history of life on Earth with a phylogenetic tree. So this is what we call a phylogeny. So the leaves of this tree, they correspond to species that we have right now at the present time. And internal nodes of this tree, they correspond to ancestral species. So we're essentially reasoning about the past. We're trying to understand, how did life on Earth actually, arise? And that's a very crucial question. And it's also, when you think about evolution, you typically think about species, but there's another key problem that is actually super important. So cancer also results from evolution. It's essentially an evolutionary process inside your own body. And when you think about a tumor, it actually starts, so I will cover this in a bit, but there's evolution going on here. So the same topics or the same concepts that apply to species phylogenetics also apply to cancer. And I will touch on this later. I actually have slides about it, so I'll just wait a little bit. And then finally, there's, you try to make sense out of the sequences, and it has to do with pattern matching. Good, so, 4 different topics, 4 different biological questions. And, this is all straight up computer science. So for sequence alignment, it's going to be dynamic programming, added distance. So if these concepts are not familiar to you, don't worry, you'll learn all of this. And this is kind of like, as you become a better computer scientist, you will also become a better computational biologist, because the same techniques that you learn in your algorithms courses, you'll be able to apply them immediately to this field. So there's a lot of stuff going on here. So there's dynamic programming, there are graphs, there are tree algorithms, some complicated data structures like suffix trees, arrays. All of that is immediately applicable to biology. Good. Now let me talk about cancer. So that's so I'm going to switch from talking about the course and the field to talking about cancer genomics, and that's pretty much what my research is about. That's what we do in a group. Well, one of the 2 things we do in a group. So let's talk about cancer. So when you think about a tumor, you can actually all tumor cells can be you can trace their lineage back to a single founder cell. And in this founder cell, what happened was a somatic mutation happened. So what that actually means is that for whatever reason, a position in the genome changed from maybe an A to a G. And because of that change, this cell actually became fitter than surrounding cells. So it generated a lot of offspring. So what happens is you get a clonal expansion. So you get many descendants. So this cell is going to divide faster than surrounding cells. And as such, because of cell division, you replicate the genome. So this mutation will be passed on to its descendants. So you get a lot of cells with this initial blue mutation. And this is called a clonal expansion. And descendants of this very first founder cell, they can gain additional mutations. And this leads to additional clonal expansions. And what you get in the end is this tumor that is, it's not homogeneous. There's a lot of heterogeneity. You have different clones inside a tumor. And this intra-tumor heterogeneity is actually what makes cancer so hard to treat. This is why you get resistance. Because if you start treating a tumor, maybe you only kill a subset of the clones, not all clones. And clones that you didn't kill, because they don't respond to treatment, they will take over. So that's really why, this is such a hard process. There's evolution happening. It's constantly changing, and it's not homogeneous. So you can actually represent this with a tree as well, just like species evolution. So the analogy would be, these clones. So clone is a group of cells with identical mutations. They correspond to different species. So they will correspond to leaves of your tree. And you try to, to model their history by a tree. And the reason why we care about phylogenies, tumor phylogenies, there's a couple of reasons. So there's basic science. So if you basically are able to reconstruct the tree of different tumors, actually, you guys need to do a little thought experiment with me. So when we look at species, life on Earth, everything is related. It's all a single evolutionary process. There's only one tree of life. However, when you look at tumors, they're actually separate evolutionary processes. So each tumor is a separate instantiation of an evolutionary process. So they're not related. So essentially, a tumor inside one person is different from a tumor inside another person, because it's 2 different separate processes. So you have different trees. Now, what you can do is you can actually start comparing these different trees in different patients, and maybe by doing this comparative analysis, you can start finding common patterns of tumor evolution. And these common patterns of tumor evolution, they will hint at biological mechanisms that are important, and they will also tell you, how do tumors evolve? What are the general rules that apply? So that's why it's actually super important to be able to do this. And the other thing that's important about tumor phylogenies is they actually allow you to reason about metastasis. So 90% of cancer patients, they die because of metastasis. So metastasis is when tumor cells leave the primary tumor, so this initial location in your body where it started, and start spreading to other locations in your body. And that's, that's bad. That's why, most people die of cancer. 90% die because of metastasis. So you want to prevent this at at all costs. So this is something that you don't want to have happening. So what you can actually do is, to understand how metastasis develops, you can kind of like do this analysis backward in time. So there's a lot of data of metastatic tumors where you get basically sequencing data from different locations inside one body. And then you could try to reconstruct the pattern of spread. In what order were these metastases seeded? And what were the clones that actually migrated from one location to another location? And why did they migrate? Is there a certain mutation that is associated with this migration? So those are the questions you can start answering. And in order to do that, you need to reconstruct the tree. And finally, there's also a clinical reason. So the tree actually gives you, it actually allows you to reason about treatment. So when you treat a tumor, you want to treat as many clones as possible. So you want to target mutations that are closer to the root, because they will be shared in a lot of descendants. So that's what a tree allows you to do. So the point that I'm making here is, these downstream analysis, they actually critically rely on accurate tumor phylogeny inference. So this task of inferring tumor phylogeny is very important because it affects a lot of downstream analyses. The three that I hinted at here. And the key challenge in phylogenetics is you're trying to reconstruct the past from data at the present time. And there's one little additional challenge in tumor phylogenetics, and it actually has to do with our data. So I'm just going to give you this very brief introduction about cancer sequencing. So when you sequence a tumor, what typically happens is, instead of just a single sample, you get two samples. So you get a sample from the tumor, that's the tumor sample, and you get a sample from normal tissue. So there's two biopsies. You have a biopsy from normal tissue, and you have a biopsy from the tumor. And then you basically sequence both biopsies. So if you remember, when you sequence, you don't get a genome from start to end. You get these short reads, 100 base pairs, which is orders of magnitude smaller than the human reference genome. So the first thing that you do is you basically take these short reads and you align them to the reference genome. So align basically means is you want to figure out this read that I have, where does it fit on the reference genome? What's the best position? And this is actually course topic number 1. So in order to answer that, you need to do sequence alignment. And once you've done that, once you have an alignment of the tumor, of reads from the tumor biopsy and reads from the normal biopsy, you can actually start comparing the normal to tumor. And by doing so, you can identify positions in the genome where you see mutations that you have in the tumor and that are absent in the normal tissue, in the normal sample. So these are mutations that are, that are unique to the tumor. So this is basically, the mutations that you want to use in your tree. So you want to do tree inference on them. Now, the key point is when you do sequencing, you actually sequence a biopsy of millions of cells. So when you get a short read, you don't know from which cell it came. So in fact, what you also don't know is whether this this yellow and green mutation actually came from the same cell. So that's the challenge. So what actually happens is, we have this mixed sample, and it's going to give us these reads, and we don't know which mutations actually go together. And you should contrast this with species phylogenetics. When you do species phylogenetics, you actually have the genomes of every individual species. They're given to you as input. Now, I told you about the analogy between cancer phylogenetics and species phylogenetics. So instead of looking at species, we're looking at clones. But the problem here is we're not given the clones. We don't know which mutations co-occur in one clone. Instead, we have this mixture problem that we need to solve. And that actually makes it very complicated. So that's kind of like the key challenge in this field. Our data, typically bulk data, that's what we have in the clinic, is essentially this mixture. of different clones that you have to deconvolve, and that leads to a lot of computer science problems. And yeah, so essentially, I alluded to this before. So in order to answer biological questions in cancer, you actually need to have precise mathematical models with clearly stated assumptions. So you need to formulate, computer science problems where you're very clear and explicit about the assumptions that you're making. And once you do this, you can actually start thinking about, cancer evolution, and then basically you can also start assessing your models. Are the models reasonable? Do they allow for reasonable interpretations? If not, you can adapt your model. But you need a separation between kind of like method for inference and the actual model that you use, the problem statement. So, problem does not equal algorithm. That's a very big, important thing that that you guys will learn about. Now, just going back to this bulk sequencing. So you can actually represent this as a matrix factorization problem. So I know this is very technical, but I just want to give you a very brief sense and just formulate 4 key questions that we try to answer in the group. So what I'm showing you here, this is a tumor. And we took 2 biopsies from the tumor, S1 and S2. And in addition, we also took a normal biopsy, which I'm not showing. And this actually leads to this matrix. So columns correspond to positions in the genome where we have a mutation that we did not have in a normal sample. And the rows correspond to the 2 biopsies. And every entry is the frequency of the mutation. So in this case, we had 5 reads. G is the mutation. That's what we didn't have in the normal. And 3 out of 5 corresponds to a frequency of 0.6. So this is going to lead to this frequency matrix over here. And then the task is to deconvolve, to factorize this frequency matrix into 2 matrices. So one is going to correspond to the tree. So this B prime corresponds to the tree. I'm not going to tell you how, but you'll learn about it if you take my course. And the other matrix tells you the composition of the sample. So it tells you, sample 1 is 20% this clone, 10% this clone, etc. Now, the key point is, these mixtures are actually averages. So if I give you averages and you try to identify the individual components and their mixture proportions, there's many different solutions you can identify. And if you remember the question or the point that I made at the start, tumor phylogenies are input to downstream analyses. Now, if there's different reconstructions I can give for the same tumor, they could actually alter conclusions in downstream analyses, including treatment. So this is actually super relevant. You want to understand the solution space. So there's a couple of questions that we're asking in the group. Can we determine the number of solutions? Oftentimes this number is huge. So then it actually becomes a question about summarizing. And in order to summarize, you want to be able to sample uniformly at random and then summarize your samples that you've taken. And also the other thing that we ask is, can we actually reduce this, this ambiguity that we have by incorporating or by doing more experiments? So this is about experiment design. Good, so there's a lot of theory. That's what this slide means. There's a lot of theory, cool theory. And this is mainly driven by Yan Yan, a PhD student in my group. The other question that is, driven by this this non-uniqueness is actually about reducing non-uniqueness. So Leah and Noorayni. So Leah is a PhD student, and Noorayni just graduated. She was a master's student in my lab. They actually did very cool work, and essentially what they did is they were designing a follow-up single-cell sequencing experiment. So single-cell sequencing is this new technology where you sequence 1 cell at a time. It's very error-prone and costly, but what they were trying to do is they were trying to identify how many cells do you need to sequence in order to select the correct tree among this solution space of trees. And that's a very cool computer science question that you can formulate. So that's what I did. So these are all, by the way, papers. So if you're interested, look at my webpage and you'll be able to read more about this. And the other thing that we started doing in the group, so Joshi also just graduated. She got her master's degree here. And she was thinking about visualizing spatial structure in tumors. So what she did is she essentially wrote like a web app using D3, where she's outlining. So basically what you see over here is a tumor where many biopsies were taken. So every grid point is a biopsy location. So they took a lot of biopsies of this tumor, and then you solve the deconvolution problem. You get a tree, and you also get kind of like the composition of the clones at every location. And you can use ISO lines to draw essentially where your different clones occur. And this is going to give you a spatial view of the tumor. So it's a very cool app. It's actually take a look at it. Yeah, it's on my webpage. So that's what she was working on. And let's see what else we have. Yeah, so I just introduced like research in my group, but there's more faculty in the computer science department. So in particular, we have Professor John Penn, who does machine learning in computational biology. So there's a lot of machine learning involved as well. And there's Professor Saurabh Sena, who does regulatory genomics. He's looking at gene expression. And Professor Penny Warnow is doing species phylogenetics. And I gave you a little introduction about that here today. And there are other faculty in CS and ECE as well that do all kinds of cool research in this area. Good. I think that's all I have for today. So I'm happy to take any questions.
What do you think the main difference between computational biology and bioengineering is?
Computational biology and bioengineering. That's a good question. Let me see.
Or if there even is a difference.
Yeah, so the question that people typically ask is about computational biology and bioinformatics. So my understanding of bioengineering, it also includes, things like tissue engineering and, manipulating biological systems. So for me personally, computational biology is more about, computational techniques to analyze data. So, and typically this data is data obtained from, high throughput sequencing. So this could be DNA sequences, RNA, protein sequence, So it's really about computational methods. And I think bioengineering also includes more experimental techniques to generate new systems or mimic existing biological systems. That would be my explanation. But I do want to allude to the difference between computational biology and bioinformatics. So these two things are actually used interchangeably. So if I go back, computational biology and bioinformatics, it's typically used interchangeably, but there is a subtle difference. And the difference is that typically bioinformatics is viewed as applying existing methods that have already been developed to analyze biological data. And computational biology is about developing new methods. So that's a very subtle distinction. But I would say it's kind of like the distinction that is actually made, that is recognized in the field. So there's a very solid difference between the two.
Okay, great. We had one question submitted here. How would computational biology be helpful for things like vaccine development?
Yeah, extremely helpful. So, for instance, let me give you an example. Let's talk about, for instance, like the mRNA vaccine that you've heard about this week. So there's two essentially. There's one from Moderna, but the one that was in the news this week is the one from Pfizer and BioNTech. So that's an mRNA vaccine. So what they actually did is they looked at the coronavirus and they identified like one protein. In this case, they were looking at the spike protein, and they tried to identify, which fragment of the genome is actually relevant that will lead to an immune response. So they essentially, identified in the genome, in the coronavirus genome, a subset of this, a small component of this spike protein, and it's very tricky because there's a couple of things that you need to do. Because the mRNA vaccine is you're just going to have the messenger RNA corresponding to this subsequence injected directly inside inside a patient. And the idea would be, this messenger RNA is going to be taken up by cells, and it will actually be transcribed. So it's going to lead to viral protein. but it's just a small piece of a bigger protein and nothing else of the virus. So it's completely, it should be harmless. That's the idea. But the key questions there are, you want to identify a piece of the genome that will lead to an immune response, because you're trying to train the immune system of the host. And there's a couple of things that you need to account for. So there's, so essentially what's happening is, I can spend a lot of time talking about this, but there are a couple of things that could go wrong. So one thing that could go wrong is that a small piece of messenger RNA that you injected actually codes for protein sequence, and this protein sequence is very likely to be cleaved by the body. So it's not going to end up being presented on the cell surface of MHC, and it won't generate an immune response. So you want to prevent that from happening. And the other thing is you want to find something that leads to an immune response in the majority of the human population. Lots of computational questions, lots of machine learning that you need to do.
Yeah, and kind of going off of that, I guess, what do you think the future of computational biology and bioinformatics looks like, maybe aside from vaccines or other applications?
I think, yeah, that's a good question. I think biology, there's no escape from computer science. So it's kind of interesting. You know what we have here at Illinois, like CS plus X. So computer science is kind of like everywhere. I would actually argue that, especially for biology, this is the case, because right now the paradigm shifted completely. It's really about generating lots of data, then interpreting it, and this interpretation requires computer science. So you need computational thinking. And this is a general trend. This is what you see in all the sciences. You need computer science for everything. So that trend will basically continue, and I don't see it changing. Essentially, biology is just becoming computational, and you need computer science. But there's different types of computer science that you need. You need to be able to run, algorithms. So there's more applied stuff, but there's also developing new algorithms, and that's closer to computer science, and that's kind of like where we are positioned here.
Okay, I think one good question to close off on here would be, for a student who would want to get involved in possible research like this or computational biology, what advice would you give them? And also, what skills would you advise them to focus on? What would they need in order to end up being in your position one day?
Focus on computer science. That's really my key advice. Like if you're strong, have a solid understanding of computer science and like the foundational algorithms and data structures, and this ability to think computationally, to essentially be able to like think abstractly, that is extremely powerful, and it will help you everywhere. So this notion of computational thinking, that's essential. And once you have that, you want to learn a little bit about the biology. You want to be able to have a conversation with biologists. You want to be able to read biological papers, biology papers, and your ability to think computationally and abstract away is going to be extremely helpful, because it allows you to kind of like identify, what are the relevant details, what are the general trends, and that's going to lead to very clean, computer science problems, and they in turn will help biologists tremendously.
Okay, thank you so much, Professor El-Kebir, for talking to us about computational biology and bioinformatics and how their applications impact different fields. And thank you everyone else for coming.
Absolutely, it was my pleasure. Thank you guys.